lovely viewers. You are all welcome to 2026 Biology WAEC Practical. What do I say? 2026 Practical Biology WAEC question. Look at what's the 2026 Biology Practical. We have been given a lot of specimen for us to dissect and use it and answer the various questions which you will be asked. Okay, this is predicted. These biology questions and answers are all predicted. Remember, my predictions are always 90% accurate. Have you watched my chemistry practical predictions? It was excellent. All right. Now we are into biology practical questions. All right, biology practical questions. A list of specimens will be displayed on the screen. Then we are not going to use it to answer our various questions. Remember, questions are being grouped into three major parts: 1, 2, and 3. All right. So we are going to use it to answer the various questions. This is question number 1. But before looking at the various questions and answer, let us look at the practical specimen. What is being given in the specimen, so that you will know as we go into it vividly. Alright, pot, granule pot. You have seen that containing seed. Specimen B, we have crushed or meal roasted granule seed. Specimen C, we are being given beaker with 20 ml of water. Specimen D, we are being given beaker with 20 ml of table salt solution. Specimen C, we are being given beaker filled with sandy soil. Specimen F, we are being given portion of elementary canal of a small mammal from esophagus to the end of the stomach. We have also a specimen G being given a twig or a branch of plant with a mixed color picture of a parasite plant on its host. Specimen H, we are being given evaporated milk. Specimen J, we are being given fresh fish. K, we are being given dry maize grain. Specimen L, are being given tomato paste on a petri dish. Specimen M, we are being given fresh tomato fruit. Specimen N, rotten tomato fruit. Specimen P, thermometer. Specimen Q, meter rule, and specimen R, quadrant. What is expected in this our specimen being given? What is expected in this our specimen being given? Now let's go into question number 1. In our deduction of the question that I'm going to ask, question number 1 says, study specimen H, K, and M and answer question number 1A to what? Question number 1C. Now, in the order of the reagent given, we are being given iodine solution, we are being given Benedict solution, and we are being given sodium hydroxide and copper 2 tetraoxosulfates. These are reagents for food tests. Iodine solution is used to test for starch. Benedict solution is used to test for reducing sugar. Sodium hydroxide and copper 2 sulfate is used to test for protein. Alright, that is why it is very, very important you see this question. That is how you should know that these are the questions that they are going to ask. Given this reagent, you should know that food test is what is common. Now let's go. Use the table below to complete the information provided. Use the table. Now what is our specimen H? Remember, go back to the screw back, you see the specimen H. Now, in the question, this test portion will be given to you. What we normally do now, this test portion will be given to you. You are only expected to answer the observation and the inference. But in case you are not given, this is how you are going to study your table and use it to answer this question perfectly. Is that understood? Now, let's go. In number 1, in question number 1, it says, add 2 drops of iodine solution on a boiled gram paste of specimen K on a white tile. Remember, specimen K is a grain of maize. you have to do what? You have to boil it first, because if you don't boil it, you cannot examine it using what? Iodine solution. Iodine solution will not break down the seed coat. Alright, so you need to boil it down and crush it if possible. Then you now add your iodine solution. When you add your iodine solution, what are you going to observe? You are going to observe a blue-black coloration. Blue-black coloration. Some people can say a dark blue coloration observed, and that confirms the presence of what? Of starch. Starch is present. Anytime iodine solution is used on a substance containing starch, a blue coloration, a dark blue coloration is what? Observed. So that should be what? This is what you are going to observe. Alright. Number 2, put small amount of H in a test tube and add 5 ml of water and shake to make a solution. Remember, this H is milk. You need to add water to it so that you can make a solution of it. When you add water, what next? You are going to add your aqueous sodium hydroxide. When you add aqueous sodium hydroxide, you shake. After shaking, you now add your copper 2 tetraoxosulfate 6. When you add your copper 2 tetraoxosulfate, what you are going to observe is either a purple or violet color. Purple or violet color. A purple and violet color signify the presence of what? Of protein. Look at purple violet color form. You are going to see a purple or violet color that is formed, and that signifies what? Protein is what? Present. Presence of protein, or protein is what? Present. That's what you're going to report. The next one is you are going to crush specimen N. This specimen N is tomato, a fresh tomato. Now this fresh tomato, why it is why I deduce this question is because Benedict solution is being used or is being included in the reagents, and the only substance that can be used to test for reducing sugar is fresh tomato fruits. So you are going to say it is a fresh tomato fruit. What you are going to do is that you need to crush that fresh tomato fruit. When you crush specimen M, which is what? A fresh tomato fruit, and add 5 ml of water and filter. There is a filter paper there. So this question, as you are seeing it, it is something that is likable. You get that? And filter it. When you filter it, put 2 ml of solution in a test tube and add 2 ml of Benedict solution. When you add 2 ml of Benedict solution, what you're going to see, what will follow is what? Is you have to boil it for what? 3 to 4 minutes. When you boil it 3 to 4 minutes, what you are going to observe is what? A brick red precipitate is formed. A brick red precipitate is formed. And that will confirm what? The presence of what? Reducing sugar. Reducing sugar. Is that understood? Now, there is one thing that I did not put here, but you have to take note. Specimen N. Specimen N is a rotten tomato. Is a rotten tomato. That one you might be asked to use a rotten tomato after this crushing of specimen N. You might be asked to use a rotten tomato and test. It will give you what? A negative result. A negative result means that after the decay process of that rotten tomato, there will be absence of this reducing sugar. Do you get that? The absence of this reducing sugar means that that rotten tomato has undergone what? A decay, which the sugar that was present, this reducing sugar that is present, have been exhausted. Do you get that? In case you are asked to carry out that one, that is what you are going to do. So the opposite of this one, here we now do what? No break red precipitate form and no reducing sugar. Now let's proceed. Number 1B. Give the name of each of the tests in 1AI to 1AII. What are the name of these various tests? This one is the test for starch. Iodine test is for the presence of what? Of starch. This test of protein, which is what? Burette test, is called burette test for what? For the presence of protein. The last one here, which is reducing sugar, it is what? Benedict test, which is for what? For the presence of reducing sugar. This is question number 1. We have extensively discussed question. Now let's look at question number 2. Question number 2, according to our prediction, remember this prediction is 90% accurate. Alright, don't play with my prediction. If you are watching for the first time, do well to subscribe to this YouTube channel. Alright, do well to subscribe to this YouTube channel and turn on your notification bell so that you will be the first person to always receive any message if a video is being posted. Now let's go. Question number 2 says, identify specimen C, D, E, J, P, and what? And R. We are asked to identify. Anytime you are asked to identify a particular specimen, what you're going to do is to write specimen A is this, specimen B is this, and here we have specimen C as what? As beaker with 20 ml of water. Specimen D is what? Beaker with 20 ml of table salt solution. Specimen E, beaker filled with sandy soil. Specimen J is fresh fish. Specimen P is thermometer. Specimen R is what? Quadrat. Alright, quadrat. Now, in our question number 2, AI. Now we are saying use P to determine and record the temperature of C and E. That is what you should be expecting. Thermometer. What do you think thermometer will be used for? What do you think thermometer will be used for? Now you are going to use this specimen P, which is thermometer, to determine the temperature of C and what? C and E. You are not going to determine. This question is coming from aquatic. All right, it's coming from water. Okay. Now to distinguish the aquatic habitat and the terrestrial habitat. Remember, C is what? Aquatic habitat. While E is what? Is terrestrial, sandy soil. Sandy soil is what? Terrestrial. Have you seen? The questions are what you should be expecting. Now, the temperature of C, which is what? 26. The temperature of C, which is what? Which is a beaker of water, is what? 26 degrees Celsius. If you measure the temperature of the water, is what? 26 degrees Celsius. And the temperature of the sand is what? 30 degrees Celsius. Are you following? 30 degrees Celsius. It means that the soil have a higher temperature than water. Is that understood? Now 2B say that state 2 uses of R in ecological studies. This one does not have any other thing that you are going to use this one for. Quadrat. Quadrat. If you are studying biology, of course, you know that they use it to quadrat. If you want to count the population of plants, now you throw a quadrat and begin to, they are small balls. Quadrat is like this. Quadrat is like this. So when you throw it now, you can use your hand and put here and begin to count the number of plant stem in it, most especially grasses. That is quadrat. Now we are now at state 2 uses of R in ecological study. What do they use R for? If you want to count the number of grasses that are in a particular place, like just carry your quadrat and place there. Use your finger, place on each of them. You now count each of the stem of the grasses. Do you understand? Now, it is used for what? Our answer here is what? Used to estimate population or the density of plant. It is used to estimate the population of plant and begin to count the grasses. What you are going to do is that place your quadrat. When you place your quadrat are in segment. You now use your finger now to be counting the number of stems in those grasses. Do you get that? That is what they use quadrat for. Number 2 is what? Is to study the distribution of plant species. To study the distribution of what? Plant species. That's what they want. So just make sure you study this question very, very well. The next question says, 2C, suggest one characteristic of J that can be investigated using C and D. This one is coming from osmosis. This one is coming from what? Osmosis. J is what? J is your fish. Now, if this fish is placed in C and placed in D, what will happen? Have you heard of hypertonic solution and hypotonic solution? Hypotonic solution is a solution containing what? Water. While hypertonic solution is a solution containing what? Salt. Hyper, high concentration of salt. Hypo, low concentration of what? Salt. Do you get that? Now, if you place this fish in this solution now, what will happen? It is the gill of the fish will absorb water. That is, the cells of the fish will absorb water and it will become what? Touch it. Do you get that? When you place go and check osmosis. That osmosis, you see isotonic, hypertonic, and what? And hypotonic. Osmosis. Now that is what you should be expecting from this question. Now the question is, J will swell as a result of what? Absorbing too much water. When you place a cell in a solution containing too much water, it will absorb water and become what? And become turgid. It is very, very good in plants. Plant will absorb water, they remain straight, they remain erect. It become what? Turgid. Do you get that? Not fish. Now, the next one now, if you place it in D, D is what? A solution with what? A table salt. The salt will absorb cells. The salt will absorb cells and it will become what? It will now shrink. Look at that person. It will shrink as a result of what? And become what? Plasmolyze. This question is SS1 work. When you place your cell in a solution containing salt, it will plasmolyze as a result of what? Like it will shrink as a result of what? The salt content will now absorb water from the cell, and the cell will now what? Shrink. It's different from hemolysis. Alright? Hemolysis is when you place your, more especially blood cell, in water, the cell will now absorb too much water and it will now what? It will now lyse. These are terms in what? In the cell and its environment. The cell and its environment. You see this kind of question there. The next one is state 2 economic importance of J to human. What are the economic importance of fish? Source of income. People who go into fishing, you do what? They harvest it and come and sell use for money. 2, source of protein. That is source of food. Do you get that? Now the next one. Okay, we have just answered. And what you should be asked to draw is what? You are going to draw and label what? Draw and label a diagram of what? Or specimen J, which is what? Fish. There are 2 things you should be expected to draw. 1, the transverse section of a tomato and fish. Those are the 2 things that you should expect to draw. Transverse section of tomato and what? And J, which is what? Fish. They are going to label it fully. Let's proceed. Going to question number 3. Question number 3 says, examine specimen A, B, F, G, M, and N. When you examine them, what are you going to see? State 4 observable differences between specimen A and B. Specimen A is what? Is groundnut pod, while specimen B is what? Is crushed groundnut seed. Now, what are you going to do? What you should be expecting is what? They are going to ask you the differences. What do you observe? This one is not something that you go and memorize. It's something that you see physically. What do you observe when you place your groundnut pod and you crush one? One, the one that is with the groundnut pod has what? Has outer seed, or you call it pod. The other one has no pod because you have crushed it. 2, is hard and solid. The groundnut pod is what? Is hard and what? Solid. Why this one is what? Soft or powdery, because you have crushed it. The next one is bigger, or you can say it's larger in size. This one is what? Is smaller. When you grind it, it's now in a fragment. Do you understand? The next one is it contains the whole intact seed. That the whole seed are intact, while this one, the seed or the cotyledon are being what? Are being broken. Do you understand? Now, we now proceed. 3b says, study specimen F carefully, name the part involved in digestion and give one function of each. What is specimen F? Specimen F is what? Alimentary canal. It says you should dissect what? Look at alimentary canal. Just go back to the list of those specimens. Now, 1, esophagus. Esophagus, also call it gullet. Alright, what is the function of the gullet? That is where food passes through what? A peristaltic movement. A peristaltic movement where food is being, after you masticate food, alright, when after the mastication of food, you now send it through your what? Esophagus. There is a movement called peristaltic movement. The function is what? Movement of food from mouth to what? To stomach. Do you get that? And now in the stomach now, stomach is for what? Digestion of what? Protein. Digestion of protein will take place there. Remember, protein is in two forms, the solid and the lipid form. Now in the lipid form, we have enzyme called renin. Renin will act on the lipid protein inside the stomach, while pepsin will act on solid protein inside the stomach. Alright? So the enzymes, if you are, in case you are asked, the enzyme that will act on the food in the stomach, what will happen? That is for what? Digestion of starch does not take place in the stomach. Is in the mouth, which is thyroid, will act on the mouth if it is starch. But in the stomach, the enzyme that act there is what? Is for protein. That is liquid and the solid protein. So we have digestion of protein and what? Secretion of what? Hydrochloric acid. Those are the two functions in the stomach. As the food gradually goes to the what? The small intestine. Remember, the small intestine is divided into 3 parts. We have the duodenum, we have the jejunum, and the ileum. Alright, all of them make up what? The small intestine. What will happen in the small intestine? We have absorption of digested food in what? In the blood. That is where absorption. There is inside the ileum. There is there is a thing called the villi. The villi are connected, in which absorption of food takes place in the villi. Large intestine. That is where what? Absorption of water takes place and reabsorption also. Do you get that? Now, this one always happen, and in the large intestine, undigested food will be what? Will be ejected out as what? As waste product. Now the next question is, examine specimen G and what? And 1. Name the mode of nutrition. Remember, specimen G is what? A mystoto, a branch of mystoto in what? In plant. Remember, mystoto is a plant parasite. Okay, mystoto is a plant parasite. Now, examine G and what? And name the mode of nutrition. Name the what? The mode of nutrition. 1. The mode of nutrition is what? Parasitic. Mystoto is a parasite. So the mode of nutrition will be what? Is parasitic. 2. Give 2 reasons for your answer. The reason for your answer is what? It has a hysteria penetrating through the host branch. They use their hysteria to penetrate through the host branch. 2. It depends on host for water and what? Mineral salt. Are you following? Now the next one is that state 1 effect on host. Of course, what will host? What will the parasite do to host? 1, it reduces the yield of the plant. Remember, this one we are looking at plant because it's a parasite of plant. It reduces the yield of plant. It makes plant not to grow well. And what? It weakens the host plants. And of course, eventually, by the time it continues, the plant will what? Will die. The plant will die. Now the next question that we didn't put here is what? Specimen M and specimen N. Though we have already discussed specimen M in our food test, but specimen N is what? Rotten tomato. Now you are going to compare rotten tomato and specimen N. Specimen M is a fresh tomato fruit, while specimen N is a rotten tomato fruit. Alright? Now, what you are going to do is that you are going to observe you are going to look at the observable differences between specimen M and specimen N, and the biological processes that take place in specimen N. What is the biological process that take place in specimen N? The biological process that take place in specimen N is what? Is decay process. And what are the microorganisms that are responsible for the decay process, which you might be asked? 1, bacteria. 2, fungi. Those are the microorganisms that will undergo what? The decay process. And the last question you will be asked in that section is you have to divide that tomato, that is specimen M, in a transverse section of it, and do what? And draw it. You are going to draw specimen M. You have to divide it and draw it. Then you might be asked, what type of placentation is specimen M? The type of placentation in specimen M is what? Is axial placentation. We have marginal placentation and we have axial placentation. If they now ask you which other crop that have the same, or which other type of plant that have the same placentation with specimen M, we have orange. Orange have what? Axial placentation. We have pepper. Pepper have what? Axial placentation. Do you get that? Now, for more of this video, if you are watching for the first time, I am Ojemite. For more of this video, do well to like, importantly, share, subscribe, and turn on your notification bell. Thank you and God bless you. Bye. I wish you success in your exam.